Okay, yeah, as Brett said, I've been working on soil organic matter for quite a long time, and essentially the aim of this talk is what is it and how do we build it. We could actually give it another title, and we could say, well, why, when crop residues are retained for extended periods of time, don't soil carbon levels increase quite often? And in many ways, that really was the question I was trying to answer. And I'll show you some examples of that a little bit later. This is a fairly full-on talk because, I mean, a lot of you will have heard some of the stuff I've got to say, but some of you won't. So I want to give a bit of an introduction and some looking at some of the pools that we're measuring, some of the essentially basic stuff. I'm going to try and fly through that fairly quickly. I want to go down to the role of the microbes. The role of the microbe is just crucial in this whole business, and you'll see why. And then we'll get on to some results. Now I'm going to put some, wrong one. I'll give you some lab results, but what I really want to talk about today, I want to get down to that field results. I've been pushing results through in the last few, well, ever since Christmas, trying to get results so I can put some new results in front of you people. Well, also because I wanted to look at it myself and say, is it actually bloody working? So that's where I want to go today. So some of it's going to be fairly fast and we'll just go through it, but I want to leave time for questions. Okay, so lots of carbon in the soils. Now, a lot of people will have seen this before, but so everything, living thing on the surface of this planet, not the oceans, but the land, so every tree, every blade of grass, every giraffe, person, there's about 500 billion tonnes of carbon. In the atmosphere is about 700 billion tonnes of carbon. In the top 1 metre of soil, there is 1,500 billion tonnes of carbon. So soil is a huge pool of carbon, bigger than those two put together. And you know, the general public out there have absolutely got no idea about that whatsoever. When I chat to my neighbours who I'm having a beer at Christmas time and whatever, and they say, well, is there any carbon in soil? Just got no idea whatsoever. Okay, so if there is lots there, it also can be said that there are lots has been lost. Since mechanised agriculture began about 200 years ago, about 50% of carbon in agricultural soils has been lost. And so, using pretty simple logic, if it's been lost, you would think that with proper management, and whatever proper is, we should be able to put it back. Losing it leads to soil degradation and loss of soil fertility. If we can put it back, we can improve soil quality and also help climate change, mitigate climate change, if you believe in climate change. I'm not going down that track today. Okay, so where is this soil carbon? Now, you notice there I'm saying soil carbon, not soil organic matter. When you take a soil sample, the first thing you can see, generally speaking, is bits of root, bits of straw, bits of leaf, whatever. That is plant material. It contains quite a lot of carbon. It is not soil organic matter. I'll show you something in a minute. We've got the stuff that's living, so the insects in the soil, the fungi and the bacteria, that also contains carbon. Again, that generally speaking is not considered part of the soil organic matter. Soil organic matter, well, even here I'm still saying humus, you'll see something in a minute, is what soil organic matter is. It's the passive stuff. Here I say this is dead but active. What I mean by that is, yes, it is dead, but two, it can decompose fairly quickly. This stuff is very dead, and it doesn't decompose very quickly at all. Now, just as a bit of justification, Soil Science Society of America defines soil organic matter as the organic fraction of the soil exclusive of undecayed plant and animal residues, and is considered as synonymous with humus. So that particular society, pretty prestigious soil society, and I have got exactly the same view on that. However, having said that, I'm publishing a paper in Soil Biology and Biochemistry in a couple of weeks' time. It's a European journal. When the paper went over to be reviewed, it came back and it said, you cannot use the word humus. And the reason they said I couldn't use the word humus is humus by definition almost is meant to last approximately 100 years in your soil. And they said, you can't prove the stuff that you're making is going to last 100 years. And they're right, because I'm not going to be around in 100 years. And so they said, you cannot use the word humus. And there are other terms people use. People say the passive fraction, meaning it doesn't decompose very easily. Or they call it the heavy fraction. If you shove the soil in water, the humus material will sink, the plant material will float, hence the heavy fraction. I call it the fine fraction. The reason I call it the fine fraction is because I've got a sieving method and a density, what I would call a density fractionation method. Very simple, very complicated terms for a very simple thing, but that's what I call it. And when I say density fractionation method, it's a bit like cleaning wheat seeds. You take wheat seeds and you've got some wind blowing through it, you flow the wheat seeds up in the air, the crap blows to one side. That's essentially what I do with my soil. Slightly more sophisticated than that, but not much more sophisticated. I essentially blow the organic matter away from the soil and leave the soil behind. And at the end of the day, what I end up with is the fine fraction soil organic matter, or what sometimes I'm probably still going to slip and call it humus. So basically you can pick whichever term you like, it all does mean the same thing. Okay, how do we increase soil organic matter levels or humus carbon levels? Now, in the absence of large amounts of compost or manures, the most common way is to retain crop residues and reduce tillage. Let's have a look at that. Here's a paper by a guy called Lau, published in 2010. He compared no-till and conventional cultivation at 69 paired sites across the world. There was no difference in soil carbon levels between the two treatments. Now, people write lots of papers saying reduced tillage will increase the soil carbon levels. There was a really nice paper out there, I haven't got the reference there, but what this guy showed was essentially it was a sampling error. If you only sample the surface, and as you see there, it says, although there was an altered vertical distribution, as in there was more carbon near the surface with no-till, there was less lower down. So if you only sample the surface, and a lot of people only sample 0 to 10, then you are going to see more carbon. But if you sample a metre or more than a metre, then basically you're not going to see any difference in most cases. And so the idea is that people can actually sample 0 to 10 centimetres and then try and tell you something about your whole paddock. It's just complete bullshit. People are just not doing their jobs properly. This one here, this lady here, I know she's an absolutely excellent scientist. She compared wheat stubble burning and retention in France for 31 years. Now, if you're burning stubble in one paddock and you're retaining it in another paddock for 31 years, there is a huge difference in the amount of carbon being returned to the soil, but she found no difference in soil carbon levels. And that paper was one that really did inspire me. I mean, how can you keep stubble in one paddock for 31 years, burn it in another, and get no difference in soil carbon level? In many ways, that was what I was trying to answer. That's the question I was trying to answer. Now, John Kierkegaard has got a site at Harden. That's my main field site at the moment. It's looking at different cultivation, minimum till and different cultivations compared with different stubble management methods. It's been going for more than 22 years now. In all treatments, the soil carbon levels have gone down. Every single treatment. Except mine now, I must admit, but we'll see about that. And his best yields always come from stubble burn with conventional cultivation. Always. Every single year. Now, bottom line is you guys are trying to grow crops. So, the idea that you're going to keep the stubble because it might increase your profits, that just doesn't follow from what he's been finding. But I do say he hasn't been doing his incorporations properly. But anyway, we'll get on to that too. So, in the absence of compost and manures, it seems that increasing soil organic matter levels is not as simple as retaining crop residues and or reducing tillage. So what else do we do? Big take-home message. Soil organic matter and carbon are not the same thing. We measure soil carbon because we want to estimate soil organic matter. When I was doing soil science at Adelaide Uni back in the 60s, one of the first measurements we did on soil, we measured the soil carbon levels, but we were told very clearly, you're measuring carbon because you are trying to estimate organic matter. That's what you're trying to do. Carbon is that black stuff, looks like charcoal, soot. That doesn't do very much of anything for your soil. The organic matter does. It just happens to have a lot of carbon in it, and carbon is very, very easy to measure. That's why we measure the carbon, because we're actually trying to estimate soil organic matter. I estimate fine fraction soil organic matter by measuring fine fraction carbon. That's all it is. Organic matter always contains other elements as well as carbon, and the question is, what are the elements and how much? So, soil organic matter is just dead bits and pieces of organisms. That's what it is. And if you want to figure out, well, what elements does it contain, a good place to start is by looking at the elemental composition of live organisms. So, I've shown this table many times, but it is an excellent table. Mind you, I can say the first four columns I didn't put together. That came straight out of a paper. I only did the last one, so I can't take a lot of credit. But what it does show you is 98 to 99% of these organisms, and this is true for every organism on this planet, are just 6 elements: carbon, nitrogen, phosphorus, sulfur, hydrogen, and oxygen. We've got small amounts of bits and pieces. For instance, in this human being here, the reason this one's only kind of 98% and the others around about 99% is because we've got bones and we're about 2% calcium. But these other things without bones, we're talking 99 or 99 plus, just with those 6 elements. So, for each organism, if we disregard the hydrogen and the oxygen, because I'm not really interested in that, and that's largely the water component, not only do carbon, nitrogen, phosphorus and sulfur contribute most of the mass of these organisms, but the ratio is the same. It's constant. Now, I look at that as a bit like building a house. If you're trying to build a house, you've got bricks, you've got sand, you've got lime, you've got cement. If you don't have the right proportions, you cannot build a house. If you've got 1 million bricks and 1 gram of cement, you aren't going to build much of a house. If you've got 1 million tonnes of cement but 1 brick, you're still not going to build a house. You need all the components and you need them in the right ratios. This is exactly the same. It's no different whatsoever. Okay, so is it the same for soil organic matter? We analysed this by testing stuff. In fact, this started actually down here at Hamilton. I had a field site there, and one day we were having a field day there, talking to farmers, and I felt like I wasn't really getting across to them very well. And so I said to them, what I want you guys to do is to go out into your paddock somewhere, or your farm, anywhere whatsoever, send me soil samples, and I will analyse it for you. And we'll have a look at how much carbon, nitrogen, phosphorus and sulphur. They did it, then I got people in Western Australia to do it, I got people in Victoria to do it, and I got people in New South Wales to do it. But it actually started at Hamilton. I'm going to just show you the basic results because I want to get onto that field stuff. But what I've got here is just two sets of the results. Now here we're already up to 761 soils from every continent on the planet apart from Antarctica, and we're now up to 105 Australian soils collected from 4 of the 5 mainland states. There's carbon down there, there's nitrogen. As the carbon goes up, the nitrogen goes up. This is carbon, this is sulphur. As the carbon goes up, the sulphur goes up. It's the proportion, that line there is pretty much a straight line. The proportions are constant, no matter where you go in the world. It's a fundamental truth about this humus or fine fraction material. It's got constant proportions of carbon, nitrogen, phosphorus and sulphur. But basically, we're looking at for every 10,000 units of carbon, 900 of nitrogen, close on 200 of phosphorus, 140 of sulphur. You must have all those building blocks. I mean, on my site at Harden at the moment, something I haven't seen for a while, thank goodness, is I've got a dead sheep sitting there. Now, when you guys take a baby lamb, or I guess all lambs are babies, but a baby lamb or a baby cow, and you start feeding it, you are feeding it plant material. But the cow is growing. It is transforming plant material into cow material, or the sheep is transforming plant material into sheep material. That's what it means by transformation. It's not some magical thing, and it's the same with us. We are growing. Unfortunately, some of us a bit too much, but we grow because we eat plant material, a lot of it, and we transform it into human material. That sheep is a really good example of what's happening. That sheep was fed plant material, it grew into a fully grown sheep, and then for whatever reason it died in the paddock. This happened more than, probably about 18 months ago now, and yet there is still a pile of wool and skin and bone sitting there. So yes, a lot of the material that sheep was made out of was not recalcitrant. It decomposed very easily, but there's a lot of material that's still sitting there that is recalcitrant. It's the same thing with microbes. Some of the stuff that got transformed into microbes will disappear quickly in the soil, but there is a whole heap of recalcitrant stuff that becomes humus. That's what humus is, and that's what we're trying to do. So the idea is, it would seem to get more fine fraction soil organic matter, what we need to do is to get more dead microbes. And it's pretty obvious, if you want to get more dead microbes, first you must get more live microbes. Problem is, microbes are very nutrient rich compared to the stuff that they actually eat. There's some wheat straw. For every 10,000 units of carbon, 170 units of nitrogen, 16 of phosphorus, 17 of sulphur. There's the microbe. 10,000 units of carbon, more, basically 10 times as much nitrogen, a zillion times more phosphorus, and a heap more sulphur. What it means is, if I put 10,000 units of wheat carbon into the soil, I cannot get 10,000 units of microbe carbon because I don't have enough nitrogen, phosphorus and sulphur. I'm going to be limited, especially by the sulphur and the phosphorus. It isn't rocket science. That's all I'm trying to do. I'm trying to grow as many microbes as possible from a given input of stubble. That's all I'm trying to do. And what I'm intending to do is to actually improve the quality of the food supply. So I had a hypothesis. Conversion of crop residues to microbial biomass may be limited by nutrient availability in the soil and not just carbon inputs. Essentially what I did was I took some soils and I incubated them with stubble with and without extra nutrients. The extra nutrients is just improving the quality of the stubble. That's all it's doing. So 4 soils, that's what I added per 10 tonnes of straw. Don't worry about those numbers, we'll come back to them shortly. Let's look at some results. So here we have Hamilton soil, just down the road, Harden, Buntine in Western Australia, Leeton in the Griffith area. In these 2 soils I had 3 treatments, all of them I had a control, so I added 10 tonnes of straw, but no extra nutrients. The red bars I added 1 set of nutrients, the numbers I showed you. In the Harden soil I added 2 sets of nutrients, and this is how many microbes I got. With just straw, you can see actually those green bars apart from that one are all pretty similar. Very different soils. That Leeton soil has got 65% clay in the surface. That Buntine soil has got 5% clay in the surface. Very different soils, and yet putting the same amount of straw, get much the same amount of microbes. That Buntine soil has got 0.5% carbon. That Hamilton soil had 3% carbon. Now, what you would think would be a much richer soil, it's got kind of 6 times as much carbon in to start with, and yet the number of microbes from the same input of carbon is much the same again. And so it's, so for the given amount of carbon, I'm getting much the same amount of microbes coming out of it. Improved food quality, by adding some extra nutrients to that straw, I get a lot more microbes. These two responded better than these two. We'll talk about maybe why if we've got time later. When I've got adding double the amount of nutrients to these two straws, it went up again. So by increasing the stubble quality, by improving stubble quality, not the amount, I got more microbes. And if we look at these two graphs, that's the graph I just showed you, the microbial biomass, essentially microbe numbers. That is the increase in humus. You can see how similar that graph is to that graph. So by increasing my microbe numbers, I increase my humus. And that's essentially what I was trying to do. That was the hypothesis. If I make more live microbes, I will get more dead microbes, and dead microbes equal humus. And there's the proof. That's a laboratory statement. Okay, so improving food microbe quality, not only led to an increase in microbe numbers, but to an increase in fine fraction carbon as well. Now just a couple of pickies. Before we go into that, now this is the hardened soil, so I'm saying you have to have those nutrients always together. That's how the carbon went up in that hardened soil. No nutrients, one set of nutrients, two sets of nutrients. The carbon went up, so did the nitrogen, so did the phosphorus, so did the sulfur. They were always linked together. They're going up into proportions, pretty constant proportions. So to build fine fraction carbon, you must have the nitrogen, phosphorus and sulfur as well. There's actually zillions of tons of carbon in that stubble. There's just not enough nitrogen, phosphorus and sulfur. So a couple of pickies. Fortunately that was turned out a bit dark, but this is the Buntine, so this is a Western Australian sand. You can see the sand grains pretty easily. One of the things you can see is, this is after 2 incubation cycles, you can see there's a heck of a lot of straw still there. It didn't decompose. Same amount of straw, same amount of soil, same moisture condition, same temperature condition, but there's a heap there that just didn't decompose. You add the nutrients to it, and basically it's all gone. So not only does it lead to increased carbon levels, or humus carbon level, it increases the stubble breakdown rate. And that's really important. When I go out and look in the field, if I don't see the stubble breaking down very quickly, I start thinking straight away, something's wrong, it's not working. The faster it breaks down, the better it is, as far as making humus is concerned. If we have a look at a close-up of this is the one without the nutrients, you can see the sand grain still very clear. There's a bit of straw there, there's not much activity going on that thing. There's another bit of straw down there, again not much activity. This one's got a fair bit, all that black stuff is fungal spores, so there is some activity going on there. But if we look at the ones with nutrients, you can see all the sand particles are now sticking together. It's sticking together with humus. And you can see that one bit of straw that's still left, it's well and truly being attacked by microbes. Almost gone, it's very dark. So I just wanted to show you some pickies to just say that's the kind of difference we're getting. So we're seeing a change in soil structure. It's one of the big things we're aiming for. If we improve soil structure, can we improve the farming system? In the bottom line, that is what we're trying to do. I'm not really interested in putting carbon in soils if it doesn't improve the farming structure. That's what we're trying to do. Improve the farming system and get more dollars. Okay, I'm not going to talk about that one at all. We had a second incubation study. In many ways, the most exciting science was in this one, but I'm not going to go into it today. I want to get into the field trial. So I say John Kirkby established a site at Harden over 20 years ago. It's got a combination of stubble and cultivation treatments. It's randomised, it's replicated trial. We've got 4 reps. There has been, until I came along, there was no increase in soil carbon in any treatment. Apart from his burn one, he's been keeping stubble for over 20 years and his soil carbon levels have gone down in every single treatment. There's a picture of the site plan. It's the green ones I essentially took over 5 years ago. That's where he's been doing incorporation. Now he does incorporation different to me. I'm not going to go into the differences, but what I did, I took over those 4 green strips. Initially those strips were 6 metres wide. What I did was put a 2 metre strip down the middle where we don't grow anything anymore. And I've got 2 metres there where we grow a crop, 2 metres there where we grow a crop. I add nutrients to one lot of stubble and I don't add nutrients to the other lot of stubble. And so I've got 4 reps. And in 2010 I took some cores. I showed some of the results, but not many of them after I'd analysed those cores. But like last year, we took another set of cores, and now I've got some confirmation of what's happening, and now I'm a bit more happy about showing people where it's actually gone. So let's have a look. So that's what we added per tonne of straw. Now I'm saying there on average. The straw varies from year to year in how much nutrients it holds. I've got a little simple file I wrote for Excel where I analyse my straw, I plug the numbers in, and it spits out and it says to me, add this nutrients to your straw to get the best effect. And so if you've got a poor quality straw, this year we actually had a pretty poor quality straw, then I'll need more nutrients to get the same effect as if I've got a high quality straw to start with. So what I wanted to show here is the change in carbon after 3 years. So this is what I collected in 2010. Now, a couple of things there. First of all, carbon in tonnes per hectare across the top here. Depth here in 10 centimetre increments. We're going down to 1.6 metres. A lot of people, as I say, used to measure it 0 to 10 centimetre. Just rubbish, absolute rubbish. What you can see is the red bars, by and large, are always longer than the blue bars. That is, there's more carbon where I've been adding nutrients. There's one there that isn't. But generally, more carbon where I've been adding nutrients than where I haven't. That's the mean of all 4 of the plots. Total carbon where I've been adding nutrients, 54.6, 45.7. Same amount of stubble going into those plots, exactly the same amount of stubble. But important message here, 52% of the carbon is below 30 centimetres. Now, if farmers are going to go into carbon farming and they only measure the top 30 centimetres, they'd be missing more than half of the carbon and they wouldn't get paid for it. So that's an important point to remember. We are going to have to get our sampling strategies right to get maximum benefit from this kind of thing. So there's the carbon, there's the nitrogen, much the same kind of thing. Again, 65% of the nitrogen is below 30 centimetres. So a lot of that carbon is moving down the profile. I'm not going to go into why, we don't have time today, but the carbon and the nitrogen always go together. I could show you the numbers for the sulphur and the phosphorus. It looks exactly the same. Now, what I wanted to show you here was, this is the carbon. Now, this is for each individual replicate. So we've got the 4 blocks there, blue and the red. So red where I've been adding nutrients. What that number is there is the percentage difference between the blue and the red. My blocks are not reacting the same. There is a wide variation across the plot. And that's something that we're going to have to deal with in the real world. Paddocks are going to react differently in this corner to this corner. And so we're going to have to come up with sampling strategies to get real numbers for an average number across the whole paddock. You can see here that block 1, generally speaking, is one of the lowest, and block 4 is always the highest, always. I'm not quite sure why, but that used to be, before we fenced this area off, that used to be where the sheep used to camp most of the time. So I suspect that maybe there's just a few more nutrients there, and that's why we were increasing soil carbon levels more. It was interesting this year, the farmer actually, when he collected some grain for us, he left the gate open accidentally and the sheep got in there. Luckily they didn't eat much of my straw, but they went straight to block 4 and ate the weeds there. They didn't eat the weeds down here, they ate the weeds there. And when I analysed the weeds, these weeds here had more nutrients in them than those weeds. And so things are going to be different across the plot. We're going to have to figure out how to do that. So the results are very variable across the site. Now, why are they variable? I can give you one example of why, but there's probably more. When you take a soil sample, the first thing you do is you crush it gently and then you sieve it. Soil, the definition soil, is only the material that is less than 2 millimetres in diameter. If it's more than 2 millimetres in diameter, it's gravel, and gravel is not counted as soil in soil science. And so you sieve the gravel out and you chuck that away. When you analyse the soil, you're actually only analysing the less than 2 millimetre fraction. In block 1, I had 34% gravel. It's increasing as you go down the profile, so 1.6 metres here, and I've actually got more than 50% of the dirt was actually gravel. So on average, I've got 66% soil on that plot and 34% gravel. In block 4, I've got 19% gravel and 81% soil. So I've actually got more soil. So same area, but actually more soil. On average, I had 0.24% carbon right down to 1.6 metres, and that worked out, and that's how I got my 45 tonnes per hectare of carbon. When I looked at this block 4, it actually had the concentration of carbon was slightly less, but the total mass of carbon was actually more. And again, that's something we're going to have to, I can come to terms with it easily because I've been doing it for a long time, but people often get their results back from one of the commercial labs, and it will say this percentage carbon or this percentage carbon. It's not percent carbon that you're interested in, it is mass of carbon you're interested in, because that's the bit that people are going to get paid for. And so if you've got more soil but slightly less carbon, you could still have or slightly less concentration of carbon, you could actually have more carbon as a mass. And that's something, and the sampling methodology, some of these new projects, we're going to have to follow this very carefully. DAF is providing us a heap of money to set out some field trials. I'm sure some of you know about them. And one of the things that they do say in their methodology is people are going to have to take note of the gravel. I don't know whether people get gravel figures when they send it to a commercial laboratory. It's something I've always been taught to do, but it's something we're going to have to do. So change after 5 years. Now, this is the change after 3 years for the carbon. I just wanted to remind you, I had an analysis at every 10 centimetre increment. That's what I did. I sampled in December last year. So far, I've only analysed 7 of the different depths. So I haven't got the full analysis. So what I'm going to show you now is I'm going to compare how much carbon in those 7 depths there, sampled in 2012, compared to the same 7 depths sampled in 2010. Not the total amount sampled in 2010. But what you can see there for a start is, again, all the red bars are bigger than all the blue bars, that one not. And so what's going to happen down here, I'm not quite sure. But by and large, we've got more carbon there where we've been adding nutrients compared to where we haven't been adding nutrients. Now, again, here's the blocks. Again, very variable. Block 1 is still the lowest. Block 4 is now the highest. Now, this is the one with no supplementary nutrients. This is 2010 level. This is 2012 level. 2 of them have gone up in the last 2 years. Block 1 has gone up. Block 4 has gone up. Block 2 has actually gone down. Block 3 hasn't changed much at all. If I take a mean of that, I got 33 tonnes of carbon in 2010, I got 34 tonnes in 2012, just from those 7 depths. So I have increased my carbon by 1 tonne. What happens in the places where we've been adding the nutrients? The red bars have now all increased compared to 2010. And if we look at the average numbers, we had 37 tonnes per hectare in 2010 on just those 7 depths. Now we've got 41. So the other one, we've increased it by 1 tonne. With the nutrients, we've increased it by 4 tonnes. And if we have a look at this, this is just comparing the mean values from 2010, just for those 7 depths. We had 37.2 tonnes when we added nutrients, 33.1 when we didn't. So there was a difference of 4.1 tonnes of carbon. What we could say was that by adding nutrients, we increased the fine fraction carbon by 4.1 tonnes over 3 years. Here is 2012, again, so the red bars are all higher than the blue except for that one, but now the average, we've got 40.5 tonnes with nutrients, 34.3 without nutrients, so we've got 6.2 tonne difference. What we could say is we've increased the carbon, the fine fraction carbon levels by an extra, I repeat, 2.1 tonnes compared to this one. So 6.2 minus 4.1 gives me that 2.1. I've increased it by 2.1 tonnes over the last 2 years. Now, would I be right in saying it seems that I'm slowing down? I got an extra 4.1 tonnes there in 3 years, but I've only got an extra 2.1 tonnes there in 2 years. Is it slowing down? I think the answer is actually no. For a start, this was measured over 3 years. This was only measured over 2 years, so it's a shorter period of time. This was 3 wheat crops with 3 wheat crop stubbles. We had quite a lot of stubble. This was 1 wheat stubble and 1 canola stubble, so there wasn't much stubble in the canola crop. So in this 2-year period here, not only is it a shorter period of time, I would have been putting less stubble into the soil. So at the moment, I'm saying it isn't showing any signs of slowing down. It's still going up at much the same rate as it was before. So I'm pretty happy about that. Over 5 years, we're increasing the stubble, and the carbon levels are going up. It was going up after 3 years, it's still going up after 5 years, and doesn't appear to be slowing down. So, fine fraction soil organic matter always contains carbon, nitrogen, phosphorus and sulphur, and in constant proportions. Really important that to remember. You can put as much carbon in the soil as you want. I can tell a story about a guy at Moolameen, a farmer there that I was working with at one stage, and he said, I'm improving my soil carbon levels because I'm putting on coal dust. And I thought, whoopee-doo, but what is that doing to your crop? I've got no bloody idea. I can believe it's increasing your carbon levels, but it's certainly not increasing your humus carbon levels. It's just putting coal in the dirt. Now, whoops, back we go. Now, supplementary nutrients led to an increase in microbe numbers and faster stubble breakdown. Again, that is really the name of the game. I want more microbes from a given input in food. I got that more microbes by improving food quality. And by improving the food quality, the stubble broke down quicker. Breaking down quicker means more microbes. Supplementary nutrients led to an increase in fine fraction soil organic matter measured by measuring fine fraction carbon levels. It's not the carbon per se we're interested in it's the fine fraction soil organic matter, but we measure it by measuring fine fraction carbon, because carbon is very easy to measure. And it appears to work in the lab and in the field. So, thank you for listening.